Hello, everyone, and welcome to today's webinar, Pluripotent Stem Cell Research, Fundamentals of Cultivation and Differentiation. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Viola Stella Palladino, the Global Product Manager of Pluripotent Stem Cell Research at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Viola, you may now begin your presentation.
Thank you, Michelle, for the kind introduction. So I am really pleased to have the opportunity to give this webinar today. So today we will focus on pluripotent stem cell research, and we will go through some fundamentals of cultivation, characterization, and differentiation. But let's start with a quick introduction on stem cells. So, as you may already know, all stem cells show two key characteristics. On one hand, they have the capacity to self-renewing and produce more of the same cell type. On the other hand, when they are exposed to certain patterning factors, stem cells have the ability of differentiating into many different specialized cell types. This last characteristic can be better explained by the concept of cellular potency. Cell potency, in fact, describes the ability of the cells to differentiate into other cell types, and the more cell type a cell can differentiate into, the greater is its potency. In this webinar, we will focus on solutions for pluripotent stem cells, PSCs, that are known to be able to differentiate into all the embryonic derivatives. There are mainly two types of PSCs that can be distinguished according to their origin. So we have embryonic stem cells that are originally derived from the inner cell mass of a developing embryo, whereas differently, induced pluripotent stem cells, IPSCs, are derived from already differentiated somatic cells by cellular reprogramming. Other than this difference in the origin, embryonic stem cells and induced pluripotent stem cells are actually extremely similar in terms of cultivation, morphology, marker expression, and also experimental handling. Also, in regard of the differentiation potential, they show similar properties, and they can both give rise to cells that derive from the three embryonic lineages, so ectoderm, mesoderm, and endoderm. While working with PSCs, there are three main steps that come into play. If we aim to generate new iPSC lines directly from donors, then the first main task is the reprogramming. Independently, if we work with embryonic or induced pluripotent stem cells, they both need to be cultivated for further expansion. And finally, the key step in PSC research is the differentiation process that allows us to have a functional and appropriate cellular model that we can use then for our experiments. So as you may anticipate, each one of these main steps will require further subtasks. So let's start by having a better look to PSC culture. Before going into detail, let's see how PSCs are usually cultivated. So classically, PSCs have been cultured in presence of supportive feeder cells, such as, for example, mitotically inactivated mouse embryonic fibroblasts that support the growth of PSCs through the secretion of various extracellular molecules. Unfortunately, with this type of culture, it's not actually really possible to quantify and qualify the factors that are secreted by the feeders, and therefore it's really difficult to provide a standardized culturing condition in every experiment. So this is the reason why feeder-free cultivation systems have been developed and are now actually the predominant cultivation system in PSC research. So this type of culturing basically replaced the need of feeder by providing on one side a culturing matrix where the cells can adhere and lay, and on the other side, by providing an optimized culturing media that provides the cells with all the nutrients and the factors that would have been secreted by the feeder cells. So in the light of it, it's easy to understand how a feeder-free cultivation system should always be preferred. But what to do in case you are still using a feeder co-culture system? Well, there are actually several methods that can be used to switch from a feeder to a feeder-free condition. In fact, as you may see here on the left, the removal of the feeder cells can be done, for example, by selective adhesion that takes advantage of the preferential adherence of feeders to uncoated culture plates. Or it can be done by weaning off the feeder cells over several passages, or finally, it can be done by magnetic separation. So the first two approaches are time-consuming and do not always manage to remove all feeder cells, whereas in contrast, the magnetic isolation that targets feeder-specific markers is way faster and simple. For example, here on the right, you may see the flow analysis of PSC cultures before and after removal of the feeder cells. And as you can see, not only you can obtain a highly pure population of pluripotent stem cells, but also the whole procedure takes just about 20 minutes. So clearly, when we have switched to a feeder-free cultivation system, the media that we are going to use needs to have a more robust formulation compared to the one we could use in feeder co-culture. In general, in fact, the propagation of pluripotent stem cells requires a special attention and qualified reagents. In fact, PSCs are very, very sensitive cells, and of course, a robust and stable culture media is a fundamental requisite for a successful culture. A good medium in this case should contain factors that allow the maintenance of a pluripotent phenotype and should not contain any factor that can restrict the cellular phage. Moreover, the formulation needs to be tailored for the PSC metabolism and requirements that are slightly different to the one of differentiated cells. Last but not least, the media needs to have a stable and consistent composition every time. So this allows us not only to control the culturing condition, but also to make sure that they are always optimal. So which cellular features should a good media support? Here you may see some data obtained from PSC lines grown in StemMACS DiffBase XF. So first of all, a good media allows PSC to maintain their typical morphology. Second, a robust media must support and allow the continuous and stable expression of pluripotency associated markers during the culture. By doing so, it also allows the preservation of the cells in a pluripotent state and does not induce a bias in the differentiation capabilities of our line. As just mentioned, PSC culture have actually distinct metabolic demands and do not tolerate very well stress conditions. And this is the reason why media changes have been traditionally performed with 24-hour interval. But some robust and optimized culture media have the additional advantage of allowing a more flexible feeding schedule. So for example, here you may see some different feeding strategies that can be adopted when using StemMACS IPSC. So we can have a classical everyday feeding, or we can have every other day feeding, or finally, we can have a so-called weekend-free feeding. So no feeding for 2 consecutive days. After some days in culture, PSC will reach confluence, and they will need to be splitted in additional culture plates. And as you might already know, splitting the PSC culture is a delicate time point, and of course, specific techniques and reagents are necessary so that we can maintain a stable line. In general, it's important to keep in mind that different lines have also different growth kinetics, and therefore the splitting should always be assessed by having a look at the cells under a microscope. Generally, we can say that it's a good practice to keep culture in a subconfluent stage where we can still see the colonies being separated among each other clearly. Classical splitting techniques, so for example by trypsin addition, do not work very well with PSCs because the detachment must be really gentle so that we can maintain maximum viability and we can avoid spontaneous differentiation. There are different methods to passage PSC culture. So the cut and paste method consists in dividing the PSC colony with a needle under a microscope. And actually this method takes kind of a long time, and it can be used just if we are working with a low number of culture. Therefore, we generally do not recommend it. The other 2 methods are actually more suited when working with more than one line at a time, and they are generally quicker. So for example, splitting the culture by single cell passaging, so by creating a cell suspension, is advisable when we need to perform a cell counting before seeding, or we need to analyze the cells. These are the cases because this is a relatively more hard method and therefore is not really well suited for routine passaging. For routine passaging, we advise in fact to use reagents that allow for the so-called cluster passaging. With this method, PSCs are detached from the culture matrix as small aggregates, and thus this allows the preservation of cell-to-cell contacts that are pivotal in the maintenance of a pluripotent phenotype. How does it work? So here you can see a time course detachment of PSC colonies done using the StemMACS passaging solution. As you can see, after adding the solution, the cells will start to progressively lift from the matrix. After about 5 minutes, clusters can be completely detached from the matrix just by pipetting up and down. And if needed, the aggregates can be reduced in smaller pieces by keeping on pipetting up and down. Finally, the cell cluster can be transferred and seeded into new cultureware. So, during culture, batches of cells should always be frozen in order to have always some cellular backups. Cryopreservation is not only important as a good laboratory practice, but also for the creation of large repositories and biobanks. Here you can see some advices. So always freeze backup batches of cells at an early passage. Of course, you should always freeze just good quality cells. And finally, the more defined the freezing solution is, the more consistent your thawing efficiency will be. When using a defined freezing solution, in fact, we can expect consistent performances of the PSC culture after thawing. Here you may see an assessment of cell recovery and viability of PSC lines that were originally frozen in StemMACST Cryo-Brew freezing solution and thawed in StemMACS IPS Brew. After thawing, you can see that the cells show a high and reproducible recovery rate as well as viability, and they do not have a drastic alteration in the doubling time. Additionally, you can see here below that the PSC lines retain all their typical characteristics. So we quickly mentioned that the PSC must show some specific characteristic. Let's now look more in detail what these are and what we mean by the term PSC characterization. So PSC characterization includes all the assays that are usually performed in order to confirm the pluripotency, the quality, the identity, and the safety of a pluripotent cell line. And this is a critical point, and a comprehensive characterization is mandatory when establishing new lines and also as a good practice during propagation of already established line. So during PSC characterization, we actually want to assess, first, if the line has a typical morphology. Then, if the expression of markers is consistent with the pluripotent state. Third, if the line can differentiate into cells that derive from the 3 embryonic lineages. And finally, if the line has a normal karyotype. When the line passes all these tests, then it can be considered to be a high-quality PSC line. So let's start with the first and actually the easiest feature to assess, so the cell and colony morphology. Being a PSC extremely sensitive cells, one of the first events that happens when a line loses its characteristic is a change in morphology. An optimal PSC line should in fact grow as a homogeneous and monolayer culture, whereas the cell itself presents a scarce cytoplasm and it grows tightly packed with other cells, forming colonies that are characterized by having sharp and defined borders. Here on the right, you may see an example of PSC morphology, and we strongly advise to monitor the cellular morphology as often as possible. In fact, if the cells are cultured under suboptimal condition, or they are exposed to environmental stress, or if the line is just not a good quality one, the PSC may go an unwanted spontaneous differentiation. And here you can see the most common signs of alteration in PSC morphology. So starting from the left, you may see an example of flat outgrowing of big cells, then an example of a colony that has not really well-defined borders, and finally an example of central differentiation and loss of the monolayer growth. So what should we do if we see one of these cases in our culture? Well, the best solution would be, of course, to thaw a batch of the same line that was frozen at an earlier passage. Unfortunately, we know all that this is not always possible, and therefore an alternative approach can be to perform a positive selection of PSCs by pancreatic isolation. In fact, the PSCs that are still expressing stem cell-specific markers can be isolated with the magnetic separation. So the cells are harvested, and we are going to generate a single cell suspension. Then the cells are magnetically labeled, and in this case, the labeling is for a specific PSC marker, TRA160, and allows us to target just the cells that we want to maintain. These cells are going to be retained in the magnetic fields, and after that, they can be eluted from the column, then plated, and then we can start our culture again. As you can see from the plot on the upper part of the slide, in this way we can efficiently and quickly remove differentiated cells so that we can generate a pure and homogeneous population of PSCs for subsequent culture or for differentiation. The second characteristic that PSC needs to show is a stable and high expression of intracellular and surface markers that are well known to be associated with a pluripotent phenotype. The expression can be monitored either by immunofluorescence or by flow cytometry. We generally advise for flow cytometry since this allows not only a qualitative assessment but also quantitative evaluation of your culture. Here you may see an example of the assessment of pluripotency marker expression in one IPSC line with our multicolor panel for flow cytometry. So the antibody panel described here enables the detection of both surface and intracellular markers. The percentage of the marker expression is then plotted in the matrix below and can help us to quickly assess the phenotype of the line. So as you can see, the line on the left shows high expression, marked in red, of pluripotency markers such as CA4, TRA160, SOX2, OC3, whereas it shows a low expression of a differentiation marker in green, and marker is CD15. Conversely, on the right panel, we can see the expression profile of differentiated cell line. Let's now move to the assessment of pluripotency. So PSC, by definition, needs to be able to differentiate into cellular types that derive from the three embryonic germ layers, so endoderm, mesoderm, and ectoderm. This property can be assessed in various ways, both in vitro and in vivo. Classically, pluripotency has been tested in vivo using the teratoma assay. Teratomas are distinct type of tumors that are made up from several different types of tissue. In the assay, putative pluripotent stem cells are implanted into an immune-compromised mouse, where they will proliferate and differentiate to form a teratoma, just in the case they are pluripotent. Nevertheless, this method is, of course, long and certainly requires ethical approval, and is not really well suited for assessment in a routine way of your cultures. In these cases, the in vitro approach is to be preferred. We have two assays that are currently being used. One is the embryonic body formation assay, and the other one is the trilineage differentiation assay. The embryonic body assay is based on the fact that high-quality PSCs, when grown as a three-dimensional aggregate in the absence of a patterning factor, will spontaneously differentiate in cellular types that derive from the three embryonic germ layers. These spherical aggregates actually slightly resemble an embryo, and that's the reason why they are called embryonic bodies. The embryonic bodies are usually initially grown in suspension for about 2 weeks, and then they are plated for about 1 week. In the first phases, cells are maintained in a culturing media that is not supplemented with cytokines or small molecules, but is formulated so that the cell survival and the multiplication can be supported. So, for example, by StemMACS DiffBase XF. The final analysis of the expression of the various markers can be done either by flow cytometry or by immunofluorescence. An alternative in vitro test that can be used to quickly assess the pluripotency of a PSC line is the trilineage differentiation assay. So this test is based on the use of cytokine-containing media that allow the directed differentiation into ectodermal, mesodermal, or endodermal lineages. So if a line is a true PSC line, it will be able to differentiate. Here you may see a really schematic protocol for the StemMACS trilineage differentiation kit. So basically, one PSC clone is plated into 3 separate wells, and each one of them will be fed with a specific media from the kit consistent with one of the 3 germ layers. Already at day 7, as you can see, the cells are ready to be analyzed. Also, in this case, we can analyze the assay by classical immunocytochemistry or with flow cytometry. Here, as an example, you can see the assessment that was performed with our reaffinity recombinant antibody panel. The panel consists of 2 marker combinations for each germ layer and includes both surface and intracellular markers. This is not only a fast readout of the pluripotent potential of a line, but it also allows us to better understand if our lines are biased towards the differentiation into one specific lineage. Okay, so now that we saw how to maintain high quality PSCs, we can actually move to the core aspect of PSC research, so differentiation. So PSC differentiation is achieved by the sequential addition of patterning factors, so cytokines, small molecules, and so on, in a media formulated for the metabolic requirements of that specific cell type. The key factors during PSC differentiation are the combination of patterning factors that we add, their concentration, and of course the timing. By changing these criteria, over the last years, many different protocols have been published to differentiate different cell types, but also to differentiate the same cell type. Although, of course, this was aimed to establish an optimal reference protocol, this large amount of different possibilities creates problems in terms of experimental reproducibility. In fact, the phenotype of the cells that we can obtain with the different protocols might not always be completely comparable. So, for example, they might be in different maturation stages, and also the ratio between cells that were successfully differentiated and cells not differentiated may vary. And this leads to heterogeneous populations that are actually difficult to quantify. So other than being a confounding factor for the experimental output, the percentage among this population can also additionally vary in each round of differentiation, and this results in a poor experiment-to-experiment consistency. Therefore, there is an underlying need of solution to improve the differentiation and increase the standardization of our PSC differentiation. How can we increase the standardization of differentiation? Well, the first advice is to opt for ready-to-use media kit for the differentiation if they are available. This, of course, reduces operator errors, eliminates inconsistencies that are due to lot-to-lot variation of the reagents and of the components, and in general ease the creation of the experimental model so that we can have more time to focus on the actual experimental application and not just in generating our cellular model. So here is an example of a xeno-free and ready-to-use media kit for the differentiation of PSC-derived cardiomyocytes, the StemMACST Cryo-Brew XF. In this case, no supplemental addition of factors is required, and the differentiation is achieved by just mixing the appropriate media and feeding it to the cells. Differentiation with the StemMACST Cryo-Brew XF takes place just in 8 days. So first, the stem cells are seeded as single cells in mesoderm induction medium. Then the cardiac fate is induced on day 2, and beating cardiomyocytes can already be observed by day 6, of course, depending on the line. The differentiated cardiomyocytes can be cultured further if they are fed every second day with the StemMACST cardiac cultivation medium. After differentiation, cells show expression of several cardiomyocyte-specific markers, such as, for example, cardiac T-troponin, as assessed by flow cytometry here and also by immunofluorescence staining. The use of the media kit leads to high efficiency and, more importantly, high experiment-to-experiment consistency. As you can see here, shown by the graph, where the percentage of cells positive for cardiac troponin T in different line and experiment has been plotted. But what to do in the case we are still optimizing a differentiation protocol, or we cannot use a ready-to-use media kit? Well, we can still limit variability and increase standardization and consistency by, for example, using a ready-made xeno-free blank media as base media for the differentiation. Additionally, we of course should use high-quality cytokines and small molecules, keeping always in mind to check for their biological activity. Finally, we can use isolation strategies to ensure to have a homogeneous population of differentiated cells. In the next slides, we will present each of these topics in detail. So, ready-to-use base media. With this term, we refer to a medium that does not yet contain cytokines or small molecules for the differentiation, but it has a complex composition that helps for cell survival and proliferation. The base media is then supplemented with different combination of cytokines and small molecules, according, of course, to the differentiation pathway that we chose. Here you may see the results of early differentiation that was obtained by combining different patterning factors with StemMACS DiffBase XF. As you can see, this base media can be used directly without the need of cell adaptation and supports directed differentiation both in protocols with adherent culture and in protocols that pass by a EB stage. So how can we be sure to use always the same amount of active cytokine in every differentiation round? Well, we always should assess the biological activity so that this can be maintained constant among the experiment. This is because the cytokine-specific biological activity can depend on the vendor and on the production lot. This variation, of course, can lead to differentiation where the culture is either over or under saturated. And of course, the good researcher should assess every time that the cytokine activity is the same, or it should use cytokines where the activity values have already been measured. So, for example, with the max premium grade cytokines. Let's now come to the last point to increase standardization, that is cell isolation. So, as you know, PSC differentiation often leads to a heterogeneous mixture of cells. Part of them are correctly differentiated, and part of them have just an atypical phenotype. Having a homogeneous population is, of course, extremely important so that we can have a reliable result in our experiment and, of course, measure the response just from the correct target cells. By enriching the target cell population, we can control the percentage of PSC-derived cells so that is consistent among various experiments. Why is this important? Well, let's see this example on hepatocytes differentiation. So the cellular types descending from the endodermal lineage will pass through an intermediate stage that is known as definitive endoderm. This initial patterning can be done in vitro by the addition of specific cytokine, such as, for example, activin A. Then the sequential addition of other cytokines and small molecules will allow us for the terminal differentiation. During the differentiation, we evaluated the percentage of cells that were expressing definitive endoderm markers such as FOXA2, SOX17, and CXCR4. And what we saw was that already at this intermediate stage, the cells actually resemble a heterogeneous population. As you can see from the flow data, in fact, just about a half of the cells population does not express definitive endoderm markers. it's kind of clear that this will, of course, further reduce the success of our downstream differentiation into, for example, hepatic cells. Therefore, we modified the original protocol by including an isolation step for the definitive endoderm by using the CD184 microbit kit. Let's see how this has an impact on the final population. So by comparing the expression of hepatocyte markers in cells obtained without and with isolation step, it's clear that the latter shows a higher expression of hepatocyte marker. Importantly, of course, the same isolation strategy can be used also for the other cell types that derive from the definitive endoderm. So we are now at the end of this webinar, and we saw how pluripotent stem cells are very sensitive cells, and appropriate cell culture regions and handling are, of course, key factors in maintaining your cells at their best. Moreover, we highlighted how PSC characterization should always be performed both after reprogramming, but also as routine practice in maintenance culture. Finally, we showed several different strategies that can be used to increase the standardization during PSC differentiation. And with this, I would like to thank you all for your attention.
Thank you, Viola, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Okay, let's go ahead and get started. Our first question is, in which case would you recommend performing the trilineage differentiation assay rather than the embryoid bodies assay?
Yeah. As I mentioned, of course, both the tests are extremely valuable and efficient to assess the differentiation potential of a line. But that being said, it's clear that the trilineage differentiation assay is a faster alternative to the embryoid body assay, since it requires just 7 days compared to, about 3 weeks. And additionally, also on a handling point of view, the trilineage is way easier to perform because you just have to feed the cells with pre-made media. So I would personally suggest to use the trilineage assay as a solution when we want to do our routine assessment and we maybe are assessing different, many lines at the same time. Whereas I would suggest to use the embryoid body assay just seldomly and just in the case we are assessing just a low number of lines. So for example, when we are doing the first steps after establishment of a new IPSC line, so after reprogramming. Yeah.
Okay, your next question. What would you suggest to do in case a PSC line fails a characterization assay?
Of course, that unfortunately this can happen. So clearly, the first thing I would suggest to do is, of course, to repeat the test. If we see that the line still fails the test and additionally, for example, is starting to present morphological alteration and this additional feature, then we can be pretty sure that the line is no longer a high quality line. But before doing any other action, I would suggest to try to pinpoint what was the cause of it. So we should ask ourselves, is this loss of pluripotent characteristic due to the fact that the line somehow was not intrinsically genetically stable, or was it due to the fact that maybe it went under some stressful cultural condition? So, for example, was it split correctly? Was it fed with an appropriate schedule? Was the media correctly prepared? And so on. Once we have identified what was the variable in the culturing that changed, then we can proceed to sow a new clone that, of course, will proliferate better because we will correct this failure that we had before.
Okay, we'll move on to your next question. While assessing the expression of pluripotency-associated markers, how many should we check?
Okay, so in general, the rule of thumb here is the more markers you assess, the best it is. Anyway, in any case, we always want to make sure that we assess for a combination of both intracellular and surface markers that, of course, are well known to be associated with our pluripotent phenotype. As a minimum criterion, I would suggest at least 2 of each type. And additionally, I think it's always nice to add to the assessment also a marker for early differentiation. So, for example, we could add CD15 to our assessment.
Thank you. It looks like we have time for one more question. So, for which cell types would you suggest performing an isolation step at a progenitor stage rather than on the differentiated cell type?
Okay, so, well, generally speaking, the isolation can be performed at any stage, but of course, for some cell types and for some protocols, one time point may be better than the other, and these really depends on what cell type we are differentiating. So we generally perform an isolation step at a progenitor stage when, first of all, there is a defined progenitor stage, and when this progenitor has a well-defined phenotype, so that we can have a specific marker to label for the separation. Alternatively, we can also perform the isolation at a final maturation stage, but this is advisable just when we are working with a robust and well-defined cell type. So one example can be the PSC-derived cardiomyocyte. So yeah, in general, there are a lot of possibilities that mostly depends on what cell you are differentiating.
Thank you again, Viola. Do you have any final comments for our audience before we go today?
Yeah, of course, I would like to thank you all again for the attention, and then I would like to thank LabRoot for making this webinar possible.
Well, thank you so much for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.